Genetic screening of embryos requires sampling of the embryo. This is done by the aid of embryologists taking a very small biopsy of the developing embryo. So the embryos are cultured in the laboratory to the blastocyst stage, which is typically 5 or 6 days after fertilization. And when the embryo has reached this optimum stage of being called a blastocyst, when it is typically over 90 to 100 cells in development, the embryologist takes a very small biopsy of the outer layer of the embryo, which is going to be forming the placenta. This biopsy is tested or subjected to genetic analysis, or the cells that are biopsied from this embryo are sent off for genetic analysis, and the geneticists or the scientists studying the genetics of the embryos are going to be reporting if these cells have a normal genetic complement or not. We do know from research that almost half of the embryos that are created in any IVF lab are genetically abnormal and are not capable of forming a pregnancy or giving a pregnancy or ongoing pregnancy. A lot of the miscarriages that we get as a result of IVF are a result of genetically imbalanced embryos. So in order to optimize the outcome for a patient, if they have had multiple cycles of IVF, we would suggest that they have genetic screening of their embryos, whereby we're trying to look at if the embryo is genetically normal so that they have a higher chance of clinical pregnancy.